Let's say that we do an experiment to test the ability of different substances to digest the polysaccharide starch. In this, we add 2 milliliters of a starch solution to each of 4 tubes. And then to the first tube, we add 20 drops of water. To the second tube, we add 20 drops of the enzyme amylase. To the third tube, we add 20 drops of boiled amylase. And to the fourth tube, we add 20 drops of the enzyme pepsin. We then mix these, put them in a warm water bath, and let them sit for, say, a half an hour. If we then test for the presence of starch, now the starch was there at the beginning, and now we test for its presence at the end of the experiment. If the starch has been digested, it will no longer be there. If the starch has not been digested, the original starch will still be there. We do this by adding 2 drops of iodine. When iodine mixes with a starch solution, it turns blue-black. However, if the iodine does not have starch to bind to, then it will remain a light brown. We observe that in tube 1, the iodine turns blue-black, therefore starch is still present, so water does not digest starch. In the second tube, we observe that the iodine does not turn blue-black, it remains a light brown. The starch is no longer present, so amylase does digest starch. In the third tube, the solution turns blue-black. The boiled amylase does not digest starch. Boiling changes the shape of a protein, what we refer to as denaturing the protein, and therefore it was unable to catalyze this reaction. And pepsin, although an enzyme, is not an enzyme which breaks down starch, and so therefore starch was not digested and the solution also turned blue-black. So the only tube in which starch was digested is tube 2, which contained the enzyme amylase.